Okay, so we're going to start with the outermost layer, and then we'll work our way in. We're going to start off with the fibrous tunic, all right? Fibrous because it is made up of a dense fibrous type of connective tissue, all right? So first of all, the fibrous tunic is made up of the sclera, which is the white of your eye, and the cornea, okay, which is directly over the iris and the pupil there. All right, so when you think of sclera, think of just the white of the eye, made up of dense irregular connective tissue. And this actually provides the, I say oblong shape, but kind of like the circular shape of your eye is going to be provided by the sclera there. All right, but the nice thing about it too is because of the fact that it's made up of dense irregular connective tissue, like periosteum, right? The periosteum of your bone is made up of dense irregular connective tissue, and what attaches onto that? Muscles, tendons. All right, so that's what happens with your eye. All right, the sclera is going, the extrinsic eye muscles are going to attach onto that. So that includes the superior, inferior, medial, and lateral rectus, superior, inferior oblique, okay, and the levator palpebrae. Okay, all of those will connect to the sclera, all right, so we can move the eye around. The other component of the fibrous tunic, okay, is the cornea. All right, that's that transparent structure that sits right on top of the iris and the pupil. So they nickname it the window. All right, so there's 2 layers to it, okay, well actually, excuse me, there's 3 layers to it. The inner layer here is made up of a simple squamous. Okay, the outer layer is going to be made up of stratified squamous. And then in between, we're going to have our protein fiber, that collagen there. Okay? And because it's transparent, we're not going to have any structures, especially you're not going to have blood vessels. All right? But it's important to understand, all right, that the cornea, it does refract light. All right. So when we talk about the mechanics of vision, we are going to talk about the principle of light refraction, the bending of light rays. And that's important because that's how you're able to focus things. In fact, I'm wearing eyeglasses, and so because my lens has the inability, it doesn't focus as well as it used to, I have to wear eyeglasses to help assist with that light refraction. So we'll talk about the light refraction. But the cornea does refract light, all right? The lens is mainly responsible for most of that refraction, but the cornea does add to that. All right, so let's talk about the middle layer, the second layer, which is called the vascular tunic, okay? And this is where we're going to see, all right, meaning, you know, vessel, right? So we're going to see, all right, not just the blood vessels, and I'm excited for you guys because next semester you get to talk about the lymphatic system, but it also has the lymph vessels. I used to think of the lymphatic system as like, I don't want to call it the sewer system of the body, but that's another fluid-like, excuse me, fluid-like system, all right, helps to drain off, certainly, like if you sprain your ankle, all right, and then all that inflammation gathers in that tissue there, the lymphatics help drain that area away. It's also a great place for your white blood cells to kind of circulate around, okay? So your white blood cells can move around through the lymphatic system, but they can also obviously move around through the circulatory system, all right? So we're going to see here in the vascular tunic, we have 3 structures, the choroid, the ciliary body, and the iris, okay? So the choroid, that's that that brownish structure that we saw in the back portion of the eye there, all right? And that was what we saw that had all those nerves on it and blood vessels, all right? So we have several capillaries that are going to be found there, all right? And keep in mind, the innermost layer, the neural layer, that's the retina, all right? We have to keep all that neural tissue highly vascularized. Well, this is the layer that allows us to do that, the choroid there. All right, and it gives us that. Remember when we were labeling it, I said it looked like kind of like a brownish color to me. That's because of the melanocytes. All right, and their job, right, is light is being shown in through the pupil there. Okay, it's these melanocytes that will absorb that extra light. All right, so we don't hack or distort the image any. So one of the big important structures that allows us proper vision is going to be the ciliary body. Okay, the ciliary body is going to help manipulate the shape of the lens. All right, and so there's a couple components here to the ciliary body. Right, you have the ciliary muscles, and from the ciliary muscles you have these suspensory ligaments that attach onto the lens. And so when these muscles contract and relax, they can manipulate, all right, the lens. So when, and here's the thing now, it can be complicated, all right, but when the muscle contracts, all right, you would normally think when the muscle contracts, it's going to pull on something. But in this case, when the ciliary muscles contract, they loosen those ligaments. And when the ligaments are loosened, the lens gets fatter. Okay, more convex, so it bends more light as it's exiting out. Okay, and then when the muscles relax, the lens becomes more oblong. All right, we'll talk more about that. But keep in mind, it's the ciliary muscles that are going to alter the shape of the lens. And then also part of the ciliary body is the ciliary processes. Right, and these guys make the aqueous humor. Now remember when we were learning last week about the posterior cavity and the anterior cavity of the eye. The posterior cavity, let's see if I got up. I don't have a good one. Boo. All right, well, I guess I'm not going to be showing you. It's in the, oh, wait, here it is. Okay. Posterior cavity is this area here. And it's filled with the vitreous humor, that gelatin-like substance. The anterior cavity is all up here, all right? And that's filled with the aqueous humor, okay, which is more like the liquidy type of secretion. All right, so here's the lens. Here's the ciliary body here, all right, that's made up of the ciliary muscles. And then you have suspensory ligaments, okay? But also, we have the ciliary process, and that's the structure that helps to secrete, all right, that aqueous humor that's going to flow, all right, through the anterior cavity there. All right. Another structure of the vascular tunic, iris. This one's easy, okay? It's what actually gives your eye the color, all right, because of the melanocytes. But also in the iris, you have smooth muscle because that's going to manipulate the size of the pupil, right? We're also going to have, again, more vessels and then some neurological structures here, right? So the iris helps to divide the anterior cavity into two segments, all right? The anterior chamber and the posterior chamber. The anterior chamber is pretty much from the cornea to the iris. So, go back to this one here. All right, so here's your cornea. Okay, and so we go from the cornea back to the iris here. That's this kind of space right here. That's the anterior chamber, right there. And then the other side of the iris, all right, in this space here where you see the suspensory ligaments, all right, pretty much from the iris back here to this portion of the lens, all right, that's the posterior chamber. And this is where we're going to see, all right, the aqueous humor being produced. It's going to be produced in that posterior chamber there. I jumped ahead. Okay, so that posterior chamber is going to be between the iris and the lens. And then obviously the pupil, that's that opening there, all right, inside the iris. And that allows, all right, that communication, that connection between the posterior and the anterior chambers there. So when we're, I know I keep going back and forth, but I like this picture. Okay, so when our aqueous humor is being produced, all right, it can flow out of here from the posterior chamber, all right, into the anterior chamber. Okay, so it'll flow through. And then you got the sclerovenous sinus here. We can drain our aqueous humor off if the pressure gets too high. All right. In glaucoma, that's when we have an issue. All right. with the pressure of the aqueous humor. If it gets too high, all right. And I never understood, and I have to research this, because people joke around and say they smoke marijuana for their glaucoma, and I used to know it, but I can't tell you for detail. I know it has to do with some of the receptors here, but I can't remember which. It always used to fascinate me. People are like, oh, I got glaucoma. Smoke marijuana. All right. So let's talk about, you've heard me discuss how we affect the pupil diameter there, how the sympathetic nervous system will make the diameter larger, how the parasympathetic nervous system makes it smaller, right? And that has to do with these two muscle groups, all right? And it's easy to tell from the names of them what they do, all right? The sphincter pupillae muscles, all right, that is like a sphincter muscle. So remember the orbicularis oris muscle and orbicularis oculi, when we contracted those, they were going to make an opening smaller, all right? So your eyes will, when you squint your eyes, the orbicularis oris muscle would make that opening in your eye smaller. So the sphincter pupillae, all right, is going to constrict the pupil, all right, through the parasympathetic nervous system, but specifically cranial nerve 3, which is ocular, oh, this is awful, sorry, motor. All right, cranial nerve 3. Right, the dilator pupillae muscle, and again, you can tell by the name what it's going to do. It's going to increase or dilate the pupil, all right? And so it is innervated by the sympathetic nervous system. Okay? So when you go to your doctor and they shine a light in your eye, they're testing what we call the pupillary reflex. Okay? And so there's two cranial nerves involved in that. All right? For our reflex, it always involves a sensory portion, any type of reflex. All right? Remember the stretch reflex when they tap your knee with the reflex hammer? So you have sensory receptors that pick up the stretch, all right? And then you have a motor response. Same thing when we're talking about these types of reflexes. You have a sensory portion of it and a motor portion. And for the cranial nerves, it's always, there's two nerves usually involved, at least, okay? The first one is usually the lower number, and that's going the sensory portion of it. It just so happens to be how it is with most of these cranial nerves. Like the gag reflex I was telling you about. All right, I said that involved cranial nerve 9 and 10. So when they put the tongue to pressure and they hit you in the back of the throat, you gag. Well, glossopharyngeal is the sensory component because it has all the sensory innervation of your throat and the pharynx there. And vagus causes the motor response, the gagging part. Pupillary reflex, same thing. Your optic nerve, sensory only nerve, is going to get that light information, and then when you see the constriction, all right, of the pupil, right, that's going to be ocular motor. Okay, so 2 and 3. Okay, 2 is sensory, 3 is motor. Okay, so that's what we see here with the pupillary reflex. We shine the light in, okay, and it leads to pupillary constriction. And that's what this shows you here. All right, so here's our pupillary constriction. We shine the light, it enters into the pupil, and then the photoreceptors pick that up, and they cause the pupil to constrict. Okay, so you can see how the pupil gets smaller here. And then in a low light room, all right, like when you're in your bedroom at night, all right, and you've been lying in there for a while, okay, your pupil will enlarge itself. Or if you're walking around outside in the middle of the night, you know, not looking for something, because I'm sure you're doing it with a flashlight, but if you're wandering around dark, I wish people had a, I miss that. Oh, yeah, yeah. I don't know what color I'd want mine to be. Maybe like red. So I'd just stand there in a dark room. You could see my glowing eye, freak people out. All right. Let's get into the last layer, the retina. All right. This is known as the neural layer or the internal layer. All right. So, and we'll get into more of the details. So I'm not going to get into all the physiology behind it, but just a couple things here. The retina has a couple layers here. The pigmented layer, all right, neural layer, and then this last guy, the aura serrata. All right, we all know that a serrated edge is a jagged edge here, okay? But the pigmented layer, all right, this is that layer that's most posterior, and it sits right on top of the choroid here, all right? And it's this layer that provides retinol, which is vitamin A, for our photoreceptors, okay? Again, we'll talk more about that in lab. But this is where those excessive or extra light rays, those get absorbed. All right, so they're not bouncing around inside your eye, messing with any type of imaging that's going on in there. The neural layer, this has all the photoreceptors. Okay, so that's going to be the important talk about the photoreceptors, the rods and the cones. All right, so it's in this layer that when the light strikes it, okay, that hopefully we're going to create some sort of nerve signal or action potential. Right, and that action potential nerve signals will be generated here in this layer here, okay? And then finally, the aura serrata here, all right, this is going to be in the surrounding areas where we don't have photoreceptors, okay? It's going to be closer to the front, near the ciliary bodies here, okay? And it's just this jagged-like edge there that really, all right, there's not going to be any type of physiological importance to that region there, okay? So in the receptor, and we'll get again, and I know I'm going to sound like a broken record because we'll talk about this more in detail, all right, when we talk about the retina. But there are 3 sublayers to this. So we're going to repeat most of this in lecture. We have the photoreceptor layer. Okay, this is the outermost. And what that means is, because here's your eye, all right, light comes in, all right, and it's going to hit, all right, the photoreceptor layer last. I know that sounds weird. You would think, and again, when we go through this, you would think that the innermost layer would be the photoreceptive layer. You would think that the light would hit it, all right, but it's not going to be. In fact, let me just show you. There's the one I'm talking about. All right. So here's light, it's coming in, and it's going to hit the retina this way, okay? So this is the innermost layer here, okay? And this is the outermost layer. That's where the choroid is. So you see, we're looking at this picture here. Here's the light coming in. So you would figure, all right, that the first layer, the innermost layer, would be the photoreceptors would be right there, all right? But that's not the case. The photoreceptors are way deep in the retina here. So light has to pass through these other 2 layers. All right, you got a layer of bipolar cells and then a layer of ganglion cells. And we'll talk about their functions in lecture, I mean. All right, and then it hits, all right, the photoreceptive layer. Okay, and so that's where it gets confusing. I know it's going to be, we're going to talk about some things that will be a little bit confusing, but understand that the photoreceptive cell layer is the outermost layer, all right? That's what contains the rods and cones. The cones are for color, okay, easy to remember, color C, cone C, all right? And there's only 3 wavelengths, all right? It's got red, blue, and green, right? What are the primary colors? Red, yeah, red, blue, and green. Okay. I keep those in there. Yeah, wait. It's going to be red, blue, and yellow. Thank you. See, I always get. See, that's why my son would be ashamed of me. I should know this. Yep, red, blue, and yellow. It is. Because yellow and blue make green. And red and blue make purple. And red and yellow make orange. All right, anyways. So the cones only have the 3 primary colors. All right, and rods, all right, they're going to be the photoreceptors that operate in dim light. All right, they're going to be more useful for movement in dim light. All right. So the middle layer is called the bipolar cell layer, all right? And it's this layer that the photoreceptors, the rods and cones, are going to connect to the ganglion layer. That's the innermost layer, okay? And the ganglion layer is going to form, all right, eventually the optic nerve, because it takes all the axons from all these ganglia, all right, at the optic disc. Remember the optic disc in our eye was this circular area where all the blood vessels were going in? And I was telling you that's also the area where all the axons, right, from all the photoreceptors, well, not technically the photoreceptors, but from all the neuronal cells that are going to actually enter into that optic disc, all right, and then form the optic nerve, okay? So that's where we're going to start to generate our nerve signal, our action potentials. So I know you guys are very excited and you can't wait to find out how sight works, but we'll get there. All right, and then finally, the other two types of cells, these other interneurons, are going to be the horizontal cells and then what we call the amacrine cells. All right, the horizontal cells, all right, are going to augment or they're going to regulate signals between our photoreceptors and the bipolar cells. And then the amacrine are going to do the same thing in regards to regulation of signals between the bipolar and the ganglion cells. We'll talk about their function more. All right, so yeah, we'll skip on that. All right, a couple more things, and then we'll get into labeling here. All right, the optic disc, otherwise known as the blind spot, because there are no photoreceptors there. There's no point of having photoreceptors in that area if that's where all these blood vessels are exiting and entering into the actual eye, and also where the axons from the bipolar or the ganglion cells, all right, are moving into or exiting from the actual eye itself, okay? So this is where we're going to see all those ganglion cells, axons, are going to converge onto the optic discs and then enter out, or exit out, I should say, from the eye, all right, via the optic nerve and head back towards, all right, to the brain there, all right? The macula luta, remember the fovea centralis and the macula luta? The macula luta was this rounded structure, and at the center of it, all right, in your model, it looked like a little brown dot or yellowish dot. That's the fovea centralis. So the macula luta is this structure, all right, and then the fovea centralis is that little dot at the center, all right. But here's the important thing, and the fovea centralis is really the important part. This is where you have your best and sharpest vision, okay? Made up mainly of cones, all right, so that's going to be all the photoreceptors are going to be there. Rods are more on the periphery, all right, of the retina. Cones are going to be, all right, pretty heavily inundated in the fovea centralis, but the rods we'll see are going to be more in the periphery of the retina there, okay? And again, their main job, rods work awesome in low light, okay? So at night, that's your night vision, okay? And that's why if you take about 20 minutes to a half hour, right, in a dark, well, it's going to sound weird, in a dark room, or if you're outside in the dark, right, after about that 20 minutes, then the eye gets accustomed to those low level of light, all right, and the rods are going to be the primary photoreceptor that you'll be using at nighttime, all right? So a lot of people nickname it the night vision, okay? The lens here, again, we'll talk more about the lens in lecture, but it's the lens's job, just like in a camera, to focus, okay? Specifically, it's going to focus the light, hopefully, onto that fovea centralis, okay? So ideally, where your sharpest vision is located, that fovea centralis, that your lens is going to focus that light, or that light image of whatever you're looking at, onto the fovea centralis. Now, when things are blurry, the problem is the lens is not doing that. And that's why for someone like myself, I have these eyeglasses on because they're helping with that focusing there, okay? So keep in mind, the lens is going to be, and I'll talk more about it in lecture because I want to go off on this tangent, but I'm trying to fight myself. The lens is a pretty tough structure, right? It is made up of dense connective tissue, but also elastic tissue. And one of the problems as we get older, all right, as you know, elastic tissue, all right, starts to lose its elasticity. And so the lens cannot change shape as easily as it did in your younger years as you get older, hence why a lot of people will start to get reading glasses because the lens, all right, loses its ability. So over time, all right, it will lose its ability for light refraction, that bending of the light. And what helps to change the shape of the lens, like I was telling you, is that ciliary muscle and its attachment to those suspensory ligaments there, okay? So just a quick review, all right, when you're looking at something pretty far away, all right, 20 feet or more, okay, the ciliary muscles will relax. And when that happens, the lens flattens out. Because when the ciliary muscle relaxes, remember, I know it's kind of counterintuitive, right? You think when the muscle contracts, then it's going to yank on the ligaments and those will tighten up. But when the ciliary muscle relaxes, the suspensory ligaments, they tense up. And so they're yanking on the lens, okay? Now, when you go from looking at something far away, say it's a mile away, and then you glance down at your watch to see what time it is, all right, and you're focusing at something closer than 20 feet, that process is called accommodation. We talked about this last week, right? And when that occurs, the ciliary muscle tenses up, but that causes the ligaments to become relaxed or less tense. So the actual lens becomes fat. So I'll draw the ligaments kind of wavy because they were supposed to be relaxed. There's a picture in your book, right? And then it makes it more fatter or spherical, right? So you kind of have to abandon the thought of a muscle when it contracts, it's going to pull the ligaments to be taut and tight, and therefore it'll make it, all right, oblong. It's just the opposite. All right, vitreous humor and then the aqueous humor. All right, vitreous humor, please remember, it is in the posterior cavity. It's behind the lens. All right, it also contributes, all right, as does the choroid, to the shape of the eye. All right, and it does help with light refraction. Okay, it does slightly. Okay, so it's kind of cool, all right, that this fact here, that it's permanent fluids first produced during your formation as a living creature. All right, it's one of the few things, the remnants there, all right, that has started since your first development, all right, into becoming a human. But if you've ever had a detached retina, all right, has anyone ever heard of anyone? Yeah, I've never had one either, thank God. All right, but the vitreous humor helps to keep the retina, all right, in place, and so it doesn't detach. In the anterior cavity, we have the aqueous humor. Okay, now this is the one that's constantly being produced, all right, and its job is to support not only the lens but the inside of the cornea. All right, we talk about oxygenates it; it just provides it with oxygen. Okay, and it helps to give it the nourishment and the nutrients, all right, to undergo, all right, its functions. All right, I think, oh yeah, okay. One more, two more. I know I'm repeating myself on some of this. All right, aqueous humor production. All right, so the aqueous humor, all right, it starts off being produced in the posterior chamber. Okay, it'll circulate into the anterior chamber through the pupil. All right, and then it'll drain out of the anterior chamber through the scleral venous sinus. All right, its inability to do that, all right, causes that condition called glaucoma. And there's two different types of glaucoma, and I can't remember. One is it's close angle, and open angle, or something like that. I can't remember. But there are two different types. Okay. So the aqueous humor starts in the posterior chamber, and then it circulates into the anterior chamber, and then it drains, all right, from the anterior chamber into the scleral venous sinus there. So quick picture here, okay? All right, so the aqueous humor gets produced by the ciliary processes, which is part of the ciliary body, all right? produced here in the posterior chamber, circulates into the anterior chamber through the pupil here, and then it drains out through the scleral venous sinus. Kind of somewhat similar to the lacrimal apparatus drainage. Lacrimal gland, secretes tears, and then it gets drained across the eye into the puncta. Okay, very similar. Okay. All right. I don't know why these are out of order. I'm not going to worry about this. This is just showing you the different views. All right, and we've already seen the muscles here. All right, this is a little bit better than what our atlas has. You can see here's the superior oblique muscle attaching here into the sclera. Then you can see the inferior oblique muscle attaching onto the lateral portion here on the underside of the sclera there. And you can see it from the medial portion, right? The medial rectus, inferior rectus, and then superior rectus here. And then you can see, right, the superior oblique muscle here. There's a little trochlea there. It's a little pulley. Then if we take the eyeball out, you can see the positioning of all the muscles here, right, in the orbit. And then one quick view from the top. Okay, here you can see, all right, different muscles, and then here's the optic nerve intact. All right, let's do some labeling. I did. This one here was the ear structure. We're going to do ear on Thursday. Yep. All right, let's conclude with the eye. And, this is it, guys. The last of the slides. Kind of makes me sad a little. But this is perfect because we'll do the identification slides for the ear. And then on Thursday, when I'm going over the structures of the ear, you'll be like, oh, all right, I know what that is. So it makes it a little bit easier that way. All right, so let's start off here with the first one here, the outer portion. All right, the radar disc that sits on either side of our head, it's called the pinna, P-I-N-N-A. And basically, its job is to collect the sound, right, and funnel it into the opening here, the external auditory meatus. All right, so this is on the outside. This is the part that your glasses sit on. All right, and then it's going to transmit those sound waves into the external auditory meatus, or you can call it the external acoustic canal. Okay, either or is fine. And then those sound waves will enter in and travel down the canal and hit the tambourine that's there in the ear canal, which is referred to as the tympanic membrane. Otherwise known as, and don't write this on the test, the eardrum. The only acceptable answer is the tympanic membrane. And this is being recorded, so I have video evidence of me saying that to you people. Okay? So all of those structures make up the external ear. Okay? So the pinna, the external auditory meatus, and the tympanic membrane all make up the external ear. All right. Now we're going to go on the other side of the tympanic membrane to what's known as the middle ear. Okay? And that is made up of the eustachian tube, the auditory ossicles, which are those bones that we'll talk about here in a moment, and then the muscle called the tensor tympani muscle. You ever notice if you're flying and your ears start to pop? Okay? Well, that's the tensor tympani muscle helps to regulate or equalize pressure in your middle internal ear there. So let's start off. First structure, okay, the eustachian tube. Also, you can call it, if you want to, the pharyngotympanic duct. I don't know if you want to put that. I dared a student to do that on a test, and they did it. They actually wrote out the pharyngotympanic duct, and they spelled it correctly. I was impressed. That's what they said. I said, no, you should feel proud that you accepted my dare. All right, so this pharyngeotympanic duct, you're going to meet this later on in 211, all right, but this duct, all right, heads down into your throat, okay? And it's a common area, or it's been theorized that bacteria will creep up here and cause ear infections. And one of the things is when you are an infant, the eustachian tube or the pharyngeotympanic duct is more horizontal, like this, right? And as you age, it becomes more vertical, like it's shown here. And one of the theories is if it's more horizontal, it's easier for bacteria and whatnot to move along. But when it's vertical, it's harder. That's a theory. All right, let's do the 3 auditory ossicles. What? Hold on, I'll come right back to that. Boom. All right, the malleus. Okay, that's the first one here. Okay, this guy right here sits right against the tympanic membrane. Your tympanic membrane sits right here. So that auditory ossicle sits. All right, that's the first auditory ossicle. The second one, all right, that's the incus. That's the middle one. And then the third one, is that blank? Yes, thank you. Please tell me if it's not showing up at home. I just have to reset it. It's doing, can you see it now? Okay, just let me know if it's blank. Because sometimes we can see it here, but you guys can't see it. And then the last one is, oh, like that. Hold on. Is the stapes. And how I remember that is it looks like a staple. So what will happen is that sound will come in, all right, and it will cause these bones to vibrate. Okay, sound can travel, all right, it travels through air, it travels through liquid, and it can travel through solids. Okay, sound can't travel through a vacuum. That's why in space, since it's a vacuum, there's no noise out in space. So what happens is when these bones start to vibrate, they will amplify the sound. And we'll talk more about that in lecture. Oh, and then finally, the muscle, the tensor tympani muscle. This is the muscle if you try to pop your own ears. Okay, that's what you use to equalize the pressure inside the ear. All right, that's it for the middle ear. So let's jump into the inner ear. The inner ear is made up of the cochlea, the vestibule, and the semicircular canals. We're going to talk about all these in a moment. The cochlea, this has to do with hearing, and then these other two have to do with balance and equilibrium. Okay, so let's talk about that. Do you guys have that one in your book? Okay. We'll go through each one individually. I always think of the cochlea, that's what this is right here. Looks like a snail shell. That's what it reminds me of. All right, so here we go. Here's the first one. There's the cochlea. All right, I'm just going to flip it around, same structure. We're going to look at it from the other end. Okay? That's still the snail shell. We can just see where the nerve is going into it. All right, so I want you to think of the cochlea as a big, long tube. Okay? And it's got chambers in it, and we'll go through those chambers. Okay? So what we've done is we've just rolled that tube up, okay, to look like a snail shell. Okay? So it's got 3 different chambers in it. And now we're going to open it up. Let me skip this slide. I promise you I'll come right back to it. Okay? So here's that tube, and what we've done is, here's that long tube, and all I've done is just taken a slice out of it. Okay? So in that tube, there's 3 chambers. Okay? 1, 2, and 3. And this structure right here, That's the vestibular membrane there. See, it's good that we're identifying this, because when I go through actual how sound works and everything, it'll all make sense. All right, now let me go. All right, so what I'm going to do is I'm going to zoom in on this structure right down here. Okay, and when we zoom in, I know these slides are in a weird order. All right, this right here, these are hair cells. All right, and these hair cells are mechanoreceptors. Okay, they're a specialized type of mechanoreceptor. All right, we'll get into the mechanics of all that. All right, so there. All right. And so those hair cells sit on top of this structure. This is inside the cochlea, correct? Yes. This structure here that's in the circle there is called the spiral organ. That is actually the structure, all right, that is going to convert sound waves into action potentials, electrical impulses, okay? So we'll get into all that fun stuff. All right, so let's talk about the different chambers now, okay? The top big chamber, all right, that's the vestibular duct or the scala vestibule. And these chambers are filled with fluid. All right, not air, but fluid. All right, so that's the big duct, the big chamber on top. All right, the big chamber below it is called the tympanic duct or the scala tympani. All right, and then going back to this structure here, okay, this is our spiral organ. All right, so the thing that sits right above the spiral organ is called the tectorial membrane. And those hair cells that are here, they're attached onto that thing. All right, and then the last chamber or space that we haven't talked about, the smallest of the three, is called the cochlear duct. All right, so our spiral organ here sits on this grayish pinkish membrane here. It's called the basilar membrane. Aren't you excited to find out what all these things do? What's the purpose of all this? Don't worry. We'll talk about it. All right, that's it for the ear structures. All right, we're going to finish up here now with the last part. Okay, the vestibular system. Okay, balance, equilibrium. So we're going to start off with this structure here. All right, the vestibule, which is made up of these two structures. We'll identify those, but this is the vestibule here. All right, the utricle and the saccule. The top one is the utricle. Okay, it's this top sac here. Then the larger, darker one right below it, that's the saccule. The utricle. There. But both together, all right, make up the vestibule. All right, now I apologize for this. You guys don't have this in your book, but you have two structures here. Okay, this one that looks, and that's literally an oval right here. All right, so we call it the oval window, and we'll talk about its significance. But what sits on the oval window is this guy here, the stapes, this foot plate here, the stapes. That sits on the oval window. Okay, so when the sound waves come and hit this thing, this complex, all this starts to shake and this vibrates. And this foot plate here vibrates against the oval window and it creates fluid waves inside on the other side. We'll get into that, all right? But I just want you to know, all right, that the stapes sits right on the oval window. And then right below it is the round window. And that literally is round. That structure is a round structure, okay? We'll talk about the significance of all that in class. All right, so you notice that we have these 3 rings here, okay? And those are referred to as the semicircular canals. So this whole ring-like structure. And at the base of these rings, you see this kind of swelling like structure here? See how it kind of swells out there? That's called the ampulla of the semicircular canals. It's at the base of the rings there. So you can see it. Here's one that hasn't been dissected through. That's another ampulla. Okay. And then we have our bony labyrinth, and that includes, all right, all these rings here, okay, and then this structure over here. I tell folks it's all the parts that look kind of ivory colored. That is the bony labyrinth. So if it looks ivory colored, bony labyrinth, flip the structure around. If it looks gray, you see this gray structure here? That's the membranous labyrinth. All right, now when we're talking just about the vestibular system, just this structure over here, okay, we refer to that as the vestibular complex. That includes the semicircular canals. And remember the vestibule, which is made up of the utricle and the saccule, right? That's the vestibular complex. All right, so that's the semicircular canals. Here's the vestibule once again. All right, the top one, that's the utricle, and the bottom one, that's the saccule. Both of those together make up the vestibule. And then we can't forget about the cranial nerve that travels to this area. Cranial nerve number 8. Vestibular cochlear nerve. It branches off. The first branch that goes right to the cochlea is called the cochlear branch of the vestibular cochlear nerve. That's a lot. So if you want to write out cochlear branch of CN, cranial nerve 8, you can do that. I think that's a lot easier. What do you say? Instead of having to write out vestibular cochlear nerve, it's up to you, okay? So if you want to write in there the cochlear branch of cranial nerve 8, Roman numeral VIII, right? Go for it. And that's going to the cochlea, right? So the vestibular branch, all right, goes off over here towards the vestibular complex. Okay, so that's the vestibular branch. I have no idea why it's doing that. And that's it. Can you believe it? It's over. Now, I will send out on, what's today? Tuesday and Wednesday, I'll send out the Quizlets. All right, the remaining Quizlets for all this stuff. So, questions? Anyone want to give some words as we've concluded the new material for lab? Words of remembrance? Good times, the bad times? All right. Listen,